Chapter 6, DNA Replication, Repair, and Recombination. So we just have a little figure of replication occurring over and over again. This is how cells pass genetic information down from one generation to the next. We have a parent cell that's going to replicate its DNA and divide into two daughter cells. And then each of those daughter cells is going to replicate their DNA so that there's a molecule of DNA for each daughter. So we'll have 4 daughters and so on. Your chapter begins with figure 6.1. Differences in DNA can produce the variations that underlie the differences between individuals of the same species, even within the same family. So of course these members of the family do not look exactly the same because they do not have the same exact DNA. They share a lot in common, but they don't share 100% of the DNA. And obviously the goat and the rooster there have very different DNA. So in the cell cycle, of course, we've got interphase, which includes G1, and G2, and then we have M phase, and that's division. So to divide the cells so that we can have mitosis during development and make all of the cells of our body. And also meiosis is M phase, so that we can make the gametes that the next generation that will make the next generation. So every time we divide the cell, we have to do DNA replication if we want each cell to have the amount of DNA that it's supposed to have. And consequently, every S phase, there are spontaneous mistakes. So we can make mistakes, or the cell makes mistakes as it's replicating the DNA, which can have consequences. So Chapter 6 lesson objectives include to explain the ways DNA damage occurs and its effects on DNA replication and transcription, to compare and contrast different DNA repair mechanisms, including excision repair, non-homologous end joining, and homologous recombination. Draw and label an origin of replication, including parental and daughter strands, 5' and 3' ends, leading and lagging strands. Describe the activities of DNA polymerase, and predict the impact of leading and lagging strand synthesis on eukaryotic cell chromosomal ends. Number 6, compare and contrast eukaryotic specific problems, multiple origins, chromosome ends with prokaryotic replication. And 7, define all of the components of the replosome, DNA replication machinery. So mutations can cause changes to the amino acid sequence of a protein. A single amino acid change can cause major problems. You can review Chapter 4. That's if we change biochemical groups. So, for example, if we go from a nonpolar to a polar charged, or from a negative or positively charged R group to a non-polar group. Those would be significant changes that could cause major problems. Because why? Because the protein will fold differently, or it might fold differently anyway. An example here is that in sickle cell anemia, it's actually caused by a single DNA strand mutation. So here's a single DNA strand of normal beta globin gene. And with your studies of the hemoglobin, you should know that hemoglobin contains 2 beta globin proteins, and it includes 2 alpha globin proteins. So here we have the DNA strand that is normal, and it has an A in this position highlighted in yellow. But in a mutant strand of DNA that carries this beta globin gene, instead of an A in this position, it's a T. So we've got a single nucleotide change, a mutation, and normally that wouldn't make a big difference, but it happens to be in a key location that changes the amino acid. So your blood cells should be nice and spherical, concave. They don't have any nucleus, but they do have hemoglobin to carry your oxygen through your bloodstream throughout your body. And this shape is very important. It fits well through very narrow blood vessels, such as your capillaries. It's flexible so that it can get through. However, this single nucleotide change, which causes an amino acid change, which I'm about to show you, it really drastically changes the phenotype of the red blood cells, and you can see that they are sickled. So not only are these not able to get through the capillaries because they're not nice and round, right? They can stick in the very narrow blood vessels because they've got a different shape, but they're also really fragile and they can break easily. So together, that means that there are some extremities with very small blood vessels, like your toes and your fingers, and it could be your legs and so forth. You can get clots forming in your blood vessels, and you're going to have those clots, the cells in the clots, being very fragile, and they're going to break. So the result is you've got a lot of cells that don't get oxygenated, and that causes pain. It can also cause strokes if it's your brain that is not getting the oxygen. So this single point mutation causes a drastic change in amino acids. So instead of a glutamate, you get a valine in the protein. And what happens to the protein as a result? So here's what glutamate looks like. It has a negative charge, and valine is a nonpolar, so it's no charge. So what that means is that the protein doesn't fold correctly. We're losing a charge group that's going to help with an ionic interaction or a hydrogen bond interaction or something like that to allow folding, and now we've got a nonpolar group there that's not going to allow those sections of the protein to fold, and so it results in this devastating disease. Of course, there are advantages to the disease, but I'll talk about those in genetics. So how does a cell know what new nucleotide to put across from each nucleotide? Well, the number of H bonds is a good clue. So every time there are 3 hydrogen bonds, as shown here for cytosine, or, whoops, they're not shown on guanine. Well, we know that guanine is going to go across from cytosine, and adenine and thymine are going to have 2 hydrogen bonds, and so they're going to go across from each other. And base pairing, also the size. So we always have a purine. Let's see, maybe I can do a little drawing here. So we've got also the adenine is going to have 2 hydrogen bonds, and the guanine is going to have 3 hydrogen bonds. So we got that. And then we've also got base pairing. So we always have a purine. I'm just going to write PUR. A purine is always going to be across from a pyrimidine, and I'll just symbolize that with PYR. And that's significant because the purine is going to have 2 rings to its structure, and the pyrimidine is only going to have 1. So we've always got, when we've got the base pairing, we've got 2 plus 1 equals 3. So we've always got 3 rings going across from one phosphodiester backbone to the other phosphodiester backbone. So it's an equal 3 rings all the way down the DNA double helix. The goal of DNA replication typically is to give you identical DNA strands, DNA molecules. So for example, in mitosis, you're going to start with a parental cell here that has 2 chromosomes. You're going to duplicate those chromosomes in S phase. They're going to line up and divide and all that jazz during mitosis, so that the end result is that you're going to have 2 daughter cells with identical DNA to the parent. Okay, so we started off with a red and a blue chromosome, and we end up with 2 daughter cells that have the identical number. So that's the goal of mitosis. Meiosis has a slightly different goal, but it does go through S phase and these M phases as well. Okay, so we have a strand. The S strand is shown here in orange, and we have what is called semi-conservative replication. Parent strand is going to, those hydrogen bonds are going to break. They're going to break apart and separate. The 2 strands are going to separate. So we go from a double-stranded molecule to a single-stranded molecule. And this acts like a template, just like a piece of paper. You can scan it and get a copy of it, or you can take it to a Xerox machine and copy that piece of paper. You still have the original piece of paper, but then you've also got a copy of that piece of paper. And the copy is the new DNA strand, or S prime. S and S prime. Okay? And so we're going to get, daughter strands, daughter molecules, macromolecules of DNA to go into each one of these daughter cells when we go through cell division. So we get daughter strands. We make identical copies to pass to the daughter cells. DNA replication is semi-conservative. So we start off with 2 orange strands here, and you can see that those 2 orange strands split. We conserve the orange strands, and we use them as templates for the new strands symbolized in red color. Then the red strands and orange strands are going to split up, and you can see we've got 1 of each in each of these DNA molecules. And in this second generation, we've got all of the copies of DNA shown in green, and so forth. So we have 1 new and 1 old copy of the DNA all the time. DNA replication, semi-conservative. We're going to continue. We have DNA replication starting at what we call a replication origin. The double helical DNA is unwound at this point, and what unwinds it are initiator proteins. Initiator proteins. Now we're talking about prokaryotic replication in particular. We learned a lot about replication from studying bacteria. So initiator proteins are going to initially unwind the DNA at the replication origin, or origin of replication. We'll have single-stranded DNA templates ready for DNA synthesis. Those old orange strands are the template strands. That's the conservative part. Those are conserved. So semi-conserved, right? So we've got conserved orange strands. Each orange strand is being used as a template to for the DNA polymerase to make a new strand. So it's going to read the template and copy it. Just like, again, your scanner is going to read the piece of paper you put on it, and you're going to still have that piece of paper, but that you're also going to have a copy generated. Okay. So the newly synthesized DNA is not conserved, and it is new, and so this is why we call it semi-conservative. All right, so this is this thing here where we're replicating. We've unwound the DNA. We've unwound the DNA, and we are replicating. This is called a replication bubble. going to have to explore those. So this is a replication bubble. Can I write on this? Yes, fantastic. Replication bubble. And in the replication bubble, we have two forks that are highlighted in yellow. So we have a replication fork at each end of the replication bubble. All right, so fork. We get the word fork from like a fork in a road. So there's two of these forks. So one replication bubble for semi-conservative replication, two forks. In eukaryotes, we have more than one replication origin that initiates replication simultaneously, and that's because we have large amounts of DNA in eukaryotes. In prokaryotes, we have one single chromosome, and it's circular, so we get one origin of replication. But in eukaryotes, we have linear lines, of chromosomes, not a circle, but lines. And there's a lot more of it because, eukaryotes are a lot bigger than a prokaryote, as you've learned from studying, atoms versus prokaryotic cells versus eukaryotic organisms. When we did that in Chapter 1. And so we've got multiple origins of replication that open at the same time because you've only got so much time during your cell cycle in S phase to replicate this DNA. And all of these replication bubbles are going to run into each other eventually to complete. So we got little bubbles, they get bigger and bigger until we complete replication. And the replication is moving in both directions. Both forks are being used for replication. And so if I can get back to my pen. This is called bidirectional because it's going in both directions. Bidirectional replication. Okay. So moving on, what do cells need to make this process happen? Well, they're going to need a template strand. They're obviously going to need some nucleotides to come in here, some monomers to make the nucleic acid. They're going to need energy to catalyze the reaction, and they're going to need an enzyme to catalyze this reaction. So those are some of the things that we need. We need building blocks, nucleotides, A, T's, C's, and G's. We need enzymes like DNA polymerases. We need energy. This is going to be a delta G that is greater than 0. It's going to require energy. It's going to be endergonic or endothermic. We need other proteins, and we need, that we're going to talk about, and we're going to need template. This is the parent DNA strand. So the job of the polymerase, obviously, as you guys already know, I'm sure, is to form phosphodiester bonds between these nucleotides. So it's going to catalyze reactions that require energy. It only works in the 5' to 3' direction. It frequently falls off the DNA if left alone. It cannot unwind DNA it just doesn't, at least not in prokaryotes. Needs to add on to existing nucleic acids. It can't start from scratch. And it has proofreading activity. So here's your cartoon version of nucleotides. You'll remember that nucleotides are composed of a phosphate bound to a ribose sugar, and that makes your backbone. And then you add the nitrogenous base, such as guanine, onto it, and you have a nucleotide monomer. So we have phosphodiester bonds between the sugar phosphate groups, and the bases of the nucleotide are going to bind to each other through hydrogen bonds to the complementary strand of DNA. The DNA is only going to be made in the 5' to 3' direction. That means there always has to be a 3' hydroxyl present in order to add the next nucleotide on. The phosphate is what is the chemical difference at the 5' end. So this can be called directionality or polarity. So it's going to go in one direction, directionality, and it can be called polarity. Polarity just means that you're different, right? We talked about polarity with oxygen and hydrogen. Oops. I did not mean to hit that. And I am trying to get back to where I was. So hopefully you wrote that note. If not, you can go back and rerun this portion of the video. But the polarity just means you're different. There's a difference. And so there's this 5' end is chemically different. It has a phosphate at it compared to the 3' end, which has a hydroxyl at it. So, and it's also directionality. So please remember those two terms when referring to a DNA strand, 5' and 3'. Directionality is different, polarity is different. Hint, if you don't want to count carbons of sugar, look for the phosphate group to determine the 5' carbon. So DNA strands, right? So this is just to show you we've got a 5' carbon. Off the 5' carbon is the phosphate group. Off the 3' carbon, so here's the sugar. Here's your primary carbon, 2'. 3' is where we've got the hydroxyl group coming off. 4', and then 5' is off the sugar, and that's where the phosphate group is attached. And this is participating in the phosphodiester bond. The phosphodiester bond is shown right here in yellow. And you can see here's the 3' sugar. I'm sorry, the 3' carbon of the sugar. What used to be the hydroxyl group has now joined the phosphate group from the 5' carbon of the adjacent nucleotide, the sugar off the adjacent nucleotide. Okay, and this is how phosphodiester bonds are made. And of course, that's going to be a condensation reaction or a dehydration synthesis as well, with water being lost as we form the bond. Okay, so DNA polymerase is going to catalyze a reaction that requires energy. So we have an incoming nucleotide. It has 3 phosphate groups attached to it, so it's a deoxynucleotide triphosphate. And it's recognizing the DNA polymerase is going to recognize this 3' hydroxyl. And then it's going to add a new nucleotide on there. And as it does, it's going to cleave off these 2 pyrophosphates. So only one of the 3 phosphates is going to be retained when this nucleotide joins the growing polypeptide chain, not polypeptide, polynucleotide chain, okay, which we call nucleic acid. And this is always happening in the 5' to 3' direction of chain growth, always. And we use a template strand of DNA shown in orange to do this. DNA replication animation, please go to the HHMI video. If you have your PowerPoint, you just press in presentation mode. You can press this. It's a hyperlink out to that video. HHMI Biointeractive video number 2 is more detailed. So you can hyperlink out to that. You can take baby steps to understanding DNA replication. Self-test. Are you able to identify leading and lagging strand orientation, proteins present at the replication fork? Compare and contrast the orientation of DNA replication on leading and lagging strands. So after seeing those videos, you should be able to do that. And then this is just showing you, this is no longer cartoon style. We've got a DNA polymerase shown here in kind of a lime green. We've got the template strand shown in orange and the newly synthesized piece of DNA strand in red. And here we've got a nucleotide in red that we're about to add. This DNA polymerase is about to add to the growing polynucleotide chain. So incoming nucleotide pairs with excuse me, a base in the template strand. All right, so we've got a little in blue showing a hydrogen base pairing right there. And then the DNA polymerase catalyzes covalent bond linkage between the adjacent, then side-by-side nucleotides. Okay, so this nucleotide monomer is added to the growing polynucleotide strand with a D. And we're going to get this covalent linkage of nucleotide into the growing new strand. And what is cleaved off of that nucleotide monomer are the pyrophosphates. And then after the polymerase has done that, you can see it's got more matches to find and more nucleotides to put together to create the growing polynucleotide strand of DNA, a nucleic acid. Okay, so now we're going to move on to DNA polymerase only works in the 5' to 3' direction. So we have, how do we synthesize the other strand? We've got the old conserved strand shown in orange. And direction of fork movement is we're unwinding to the right and we're unwinding the DNA to the left. Okay. And we know that DNA is only synthesized from the 5' to 3' direction. Problem is that, you know, we've got a 5' to 3' template strand up top, and then it's in the opposite direction on the bottom. So that makes it a little bit complicated to follow the forks in both directions. We've always got to start 5' to 3', and that's how we create, why we create these what are called Okazaki fragments in the lagging strand, because of the movement of direction and the orientation of the template strand. So going down to this bottom diagram, we have the leading strand template on the left-hand fork on the top, but on the bottom we've got the lagging strand template. And then on the other side, at the other fork, we've got lagging strand template of the right-hand fork. And then on the top and on the bottom, we've got the leading strand template of the right-hand fork. So you can see that we've got two leading strands. They're diagonal. We've got two from each other, and we've got two lagging strands that are diagonally across from each other. All right, so that's how the fork looks. And just a little close-up on one of the forks. The lagging strand, we have to keep making what are called RNA primers, shown in blue, and then the DNA is the newly synthesized DNA is shown in red. And every time we unwind the DNA, we're going from left to right, and so we get more and more template. So first we had this much template, and we made an RNA strand, and then we synthesized DNA. Then we unwound the DNA and we got more of the template visible. So we had to make a second RNA primer and then synthesize more DNA off of that primer. Why do we have to make that RNA primer? Because DNA polymerase has to have that 3' hydroxyl in order to recognize it as a substrate and catalyze the reaction of making this new DNA with monomers. Okay, so we've got then on the leading strand, we don't have that problem because the direction of synthesis, 5' to 3', is following along this template that is the opposite polarity, 3' to 5'. And so as we continue to unwind, you know, we have this much and then we have that much visible. Leading strand just keeps on keeping on. It's a continuous synthesis. Leading strand is continuous synthesis. Lagging strand is going to be discontinuous synthesis, because we have to keep stopping and making these RNA primers every time the DNA is unwound. So how do we synthesize the other strand, the lagging strand? Primase adds a complementary RNA primer, as shown here in the diagram, as shown in blue. DNA polymerase replicates DNA in the 5' to 3' direction. There's DNA polymerase. So as soon as we get this 3' hydroxyl, the DNA polymerase says, aha, I can start synthesizing. Number 3, DNA ligase ligates fragments together, seals the ends, and it requires energy, ATP. And we're going to do this ATP hydrolysis to do a coupling reaction to get the energy to generate this bond. Okay, so DNA ligase is down here. Once we get the two, so we get the primer, we remove the primer. Now we've got to seal these two DNA strands together on the Okazaki fragment and join them together. So this is the lagging strand, the Okazaki fragment are what composes the lagging strand. Lagging strand ends up to be one connected copy as we continue to remove those RNA primers and replace them with DNA. Okay, but you can see we've got a problem here at the end, right? We've got an RNA primer in there, and we remove that, and there's not going to be an RNA primer in a 3' upstream of this. There's not going to be a 3' hydroxyl, so RNA polymerase is going to be lost. It's not going to know to replicate this piece of DNA, and we'll talk about that with telomerases in a second. So how do we synthesize the other strand? We've just been talking about that. We end up with DNA ligase and ATP hydrolysis. DNA ligase joins the Okazaki fragments together. And the 5', so we use an ATP. We get that on there. We break a bond to get the energy to make this other bond. We break a bond to make a bond. Break a bond to make a bond. Release the energy from ATP, use the energy to make a phosphodiester bond. So we use a 5' phosphate to ligate to this 3' hydroxyl together with the other new strand. So the two strands are put together by this phosphate that's broken off of the ATP. Frequently falls off of the DNA. So the polymerase can frequently fall off of the DNA unless it has a sliding clamp on it. So a sliding clamp keeps the DNA polymerase attached. And what unwinds everything is something called a DNA helicase, shown in yellow in this diagram. And its job is to unwind, is to break hydrogen bonds. Helicase stands for a hydrogen bonds to break. We also need to add the existing, DNA polymerase has to have an existing nucleic acid to start. So RNA primers are made to allow DNA replication to get started. Who makes them? Primase.